It's a great pleasure for me to welcome and introduce Andrej, who's here on a Indian Academy of Science Platinum Jubilee professorship, which means that he has to go around the country, and he's already done that in Mumbai, Aurangabad, Ahmednagar, and now here and later Bangalore, Mysore, etc., spreading the gospel of molecular modeling. And the reason why he is doing it is because the academy wanted the academy essentially gives these professorships to people who are experts in fields that are somewhat underrepresented in India. Jokes apart, there is no better expert than him, because he has been in this since his PhD days, where he created this piece of software called Modeler, which more than one half of the world uses to do molecular modeling, homology modeling in. But Andrej has set his sights to higher goals now, and he's looking at how to elucidate structures of large macromolecular complexes, molecular machines, etc., for which he and his lab have now created the integrated modeling platform software, right? And that is what he's going to tell you about. And I had the good fortune of spending many years outstaying my welcome in his lab many years ago, for which I'm eternally grateful. And me standing here in front of you is in no small measure because of him and his support and friendship, mentorship, etc. And I can go on all day about what a great guy he is, but I will not embarrass him any further. Except there's one thing. In the invitation, we had to choose a time at which he would come here, and he said he wants to see the monsoon. And till yesterday, not a drop of water fell on his head. And then, despite him getting soaked in Visapur Fort, he says that he hasn't seen enough of the monsoon. So please shower him with your questions during and after the talk. Andrej.
Wow, you grew up. To the professors, this illustrates why it's good to have very nice and very smart and successful students and postdocs in your lab, because then you might benefit even after they leave from the relationships you establish that way. Thank you, Madhu. And thank you for coming here today. I would like to begin by showing a model that needs no introduction. It's a model of the double helix of DNA proposed by Watson and Crick many years ago, and it's arguably the most influential model in biology and beyond. We cannot imagine biology, biotechnology, biomedicine without this model, or the consequences, implications of this model. This model illustrates very well, I think, two of the major points I'd like to underline in my own talk. The first one is that the model is best obtained by considering different types of input information. In this case, they were aware of the X-ray diffraction pattern measured by Rosalind Franklin, informing about the helicity of the system and some geometry of the system. But they also knew about the composition, stoichiometry, and chemical complementarity of the components of the system. And it was, at the very least, these considerations, perhaps something else, that allowed them to propose this model. And so, number one, as I also already indicated, to understand and modulate cellular processes, we need their models. It's not enough to just generate the data. We need to convert the data into models. And these models, as the second point, are best generated by considering all information and not just be a crystallographer or just a chemist. And so, in my talk, I will certainly get through the first two topics. I hope at least quickly, I get through the third one as well. I will begin by introducing integrative modeling of macromolecular structures, and then I'll illustrate the approach by its application to determining the structure of the NAP82 subcomplex of the nuclear pore complex. And if there's time, I'd like to generalize or broaden the integrative modeling of macromolecular structures to integrative modeling of molecular networks. The same mindset, but a different type of a system that is modeled. And to begin with, I'd like to define how I see the goal of structural biology in the technical sense, and that is to improve the accuracy, resolution or precision, completeness, and of course, for practical purposes, efficiency of structural characterization of macromolecular assemblies and the processes in which they are involved. The motivation for that is obvious. Often when we have a structural model, this model will allow us to quote-unquote understand how the system works, how it evolved, how it can be controlled or modulated, perhaps even how it can be modified or designed from scratch. And what you see on the bottom is some examples of macromolecular machines of the type that we'll be interested in. Not single proteins, not whole cells, I wish, but systems like this. There may be only a few hundred core biological processes comprising the cell, each one of which is associated with one or a few of systems on this slide. And so that kind of defines the scope of task ahead of us when we are after a comprehensive structural description of the cell at this level in its hierarchy. The mindset that we will adopt for addressing this problem has already been alluded to. Of course, to describe something most accurately, precisely, and so on, you would like to use as much information as you can possibly get about the system of interest. And so in this particular case, we want to use information from any experimental method, from any physical theory, and from any kind of statistical inference at any resolution to obtain a set or the set of quote-unquote all models that are consistent with the input information. And you can see for structural biology in particular a number of different methods here indicated with these icons, some of which are high resolution experimental methods such as X-ray crystallography. Some of them are low resolution experimental methods such as chemical cross-linking, and some of which are computational methods, whether they are physics-based, electromechanics force field based methods or bioinformatics methods. All of this provides some information about the structure of the system, and in the difficult cases, in fact, in an ad hoc manner, structural biologists have often combined one or another method in order to determine structures of really difficult systems that do not completely yield to a single method. And we just want to do this really systematically and as well as possible as computational biologists in terms of in the method development part of our effort. That being the mindset, this slide shows an actual approach that implements this mindset, at least at low resolution it shows this approach. It's a four-stage process, it's an iterative process. It begins by gathering information about the system, about the structure of the system. It's illustrated by a low resolution characteristic, a representation of the system that's being modeled. You don't compute that. You have to decide based on your experience, how are you going to represent the system? Are you going to represent it at high resolution, for example, with a point per atom, just like crystallographers invariably do, or you realize you don't have sufficient information to do that, and so you accept a lower resolution representation, such as a point per residue, or maybe even, as in this case, a point per protein. But if you have hundreds of proteins, then even such a low resolution representation will, if you can follow through, result in a model that can be helpful. In fact, there's nothing that prevents you from using different representations for different parts of the system, and even multiple representations for the same part of the system to get so-called multi-scale modeling going this way. With the representation in hand, you then go from one data set to another and express each one, or each data point, in terms of a spatial restraint, informing some spatial features such as positions, distances, the hydral angles, symmetry, shape, volume, proximity, what have you. Whatever the experiment informs is formulated in terms of a spatial restraint, which is just a function that takes a better value when the model is more consistent with that information, and a worse value when a model is less consistent with the data point or data points on which the restraint is based. You try to incorporate the uncertainty in the data, in your understanding of the data, in the formulation of the restraint as well as possible. And then, with the restraints in hand, you combine them together in some fashion, usually just the sum, weighted sum perhaps, of the individual terms into a scoring function that guides sampling, optimization, or maybe even enumeration of models that hopefully results in a set of models in the end that satisfy the individual restraints, or at least many of them, within the error bars, approximately speaking. And then such models are analyzed here for their precision, whether they are for their multiplicity, maybe there are multiple models different models that satisfy the data. Maybe there are no models that satisfy the data. What are the frustrated data that cannot be satisfied? What are the missing data that you would like to have but you don't have yet to obtain a more precise model, and so on. And this analysis will inform the iteration through these 4 stages that hopefully will result in a satisfactory model in the end at some point. So that's the process. It's a very information-centric process. That's why, and I think that is the right way to focus on when you have a difficult modeling problem on the information. You can use information to represent the system. You can use it in the scoring function. You can use it in the sampling. You can use it in filtering the sampling, and you can also use it in validation. And it's up to you to decide in any given case how you will use this information. But the more information you use, the better, the more accurate, the more precise, and more complete the model will be, and or better validated the model will be. I will go through this a little bit quickly. Since the scoring function is that incorporates the restraints based on the input information is at the heart of this approach, I'll show you this one slide and discuss it briefly about the scoring functions that we use. Very often, a scoring function will have this form. It's a sum of terms, each one of which you could call a restraint. Perhaps each one is dependent on one data point or a few data points or a data set as a whole even. And it measures a difference between a computed property of a model, such as a distance of a model you're assessing against the data point, and the measured property of the model, measured distance, NMR distance, for example. And then there's a weight as well that is added to the overall sum, to the overall scoring function, consisting of many such restraints. And when you have a scoring function like this, what you want is, in your sampling process, you want to find a model for which the difference between the computed and the measured property is 0. If you find such a model, That's it. You're consistent with your data, and you're happy. But the problem here is your weight, and does it really have to be a square function of the difference, this term, functional form-wise? That's all ad hoc, even though it has this desired property that when the distance is 0 between the two, you're happy. It's still a little bit ad hoc. To do it more rigorously, one can use instead a Bayesian scoring function, which is defined as the posterior density of the model given all information that you have available, that in turn is proportional to the product between what's called the data likelihood and some prior information, where again the data likelihood quantifies the difference between the feature of a model computed from a model and a measurement that's kind of normalized on the scale of the uncertainty through an uncertainty model, data uncertainty model that's hidden within this likelihood here. I'm not showing you. But other than that, it has a very similar behavior, that whenever the model is very likely resulting in the observed data point, it's a good model. And if it's less likely, that if this was the model that's assessed would generate the observed data point in an experiment, then that will not be a good model. Except that now we're doing this, we're combining the uncertainties in the input data points much more rigorously to get the uncertainty in the final model more accurately through this scoring function as opposed to that one. And that's all that I'm going to say about scoring functions as far as details are concerned.
When you input the weights, how is the weight, I mean, is it human defined or is it?
Here, it has to be defined based on experience, based on trial and error. That may or may not be important if you find models that satisfy the measured data points within some error estimate of each data point that's obtained independently. And then you don't worry about these weights too much. If you have to balance sacrificing these restraints versus that restraints to get the best possible model, then the value of the weight can be very important. And then you're then you're in trouble, so to speak. But in this case, you can set the uncertainties, which are equivalent to weights, either based on quantifying these uncertainties independently through replica experiments, for example, or you can make the uncertainties themselves variables in the modeling process, just like the structural coordinates are, at the cost of course, of having a worse data to parameter ratio. But the beautiful thing about the Bayesian approach is that you can compute the uncertainties just like you're computing the structural coordinates based on the information that you do have in your input. So this is a superior scoring function, but it requires more work, first theoretically and second also in the computer, because you have to, in general, sample more to get all these parameters properly estimated than you have to in this case of a least square scoring function. An example of an application of this integrative structure modeling approach is shown on this slide. First, the problem was to determine the configuration, that is the positions, of the constituent proteins, 456 of them, of 30 different types, in the nuclear pore complex, which is this ring in blue, in the middle, sitting in the nuclear envelope, shown in green, defining the gates to the nucleus for macromolecular transport in and out of the nucleus. The positions of these proteins were determined through the integrative structure modeling approach that I just described, based on information that's indicated here on this slide, on the outer rim of the slide. First, there was protein composition and stoichiometry information determined by quantitative immunoblotting for each of the 30 different types of proteins in the complex. Then we had information about the overall shape, at very low resolution, of each of these proteins from ultracentrifugation experiments and some limited sequence analysis, allowing us to represent each protein either as a sphere of a certain size or a flexible string of a small number of beads. We also knew the approximate position of each of the 30 different types of a protein in the nuclear pore complex from immunoelectron microscopy experiments that relied on gold-tagged antibodies against each of the nuclear porins tagged with protein A, separately for each nuclear porin, resulting in noisy positioning in the two coordinates of the system, the axial and radial coordinates, but nevertheless, despite the noise, informative, because they were smaller the errors than the size of the system. We also had information from low resolution electron microscopy data, like this map shown here, that clearly indicates an 8-fold symmetry in this view. There's another 2-fold symmetry in the perpendicular view. And finally, and very importantly, we had information about subsets of proteins, 5 or so, that are interacting with each other from affinity purification experiments following partly breaking apart the nuclear pore complex. So these affinity purification experiments will give us subsets of proteins that are close to each other by design of the experiment in the entire intact nuclear pore complex composed. And then, with all those restraints, or all this data, the corresponding restraints were formulated, and the simulations, or I should say sampling, was performed, starting, as illustrated in this movie here, starting with random positions of proteins in space. You don't need any knowledge to do that. And then kind of shaking and baking these proteins through a version of Monte Carlo search to find those configurations that satisfied as many of these restraints as well as possible. And because the optimization or the sampling is clearly stochastic, we had to do this a large number of times, hundreds of thousands of times independently. And it turns out that in this case, you do obtain, first of all, solutions that satisfy most of these restraints within the error bars. And second, that these solutions were quite similar to each other. And those that didn't satisfy the restraints well were different. And if you superpose these similar solutions on top of each other and define and express them in a probabilistic fashion, this ensemble of independently calculated good scoring solutions, you get a density plot that can be colored by the type of a protein that dominates a certain volume of space in that cluster of good scoring solutions. And that's this density here that is a result of integrating these different types of information, none of which clearly would be sufficient to determine the structure on its own, but when you put them all together, at least in this case, you can get a model that's at about 5 nanometers on the average precision level for each protein.
How many restraints were used here?
I'm sorry?
How many restraints were actually?
So, you can imagine, it's a little, one would have to go into much more detail to even say what a restraint exactly is. But you can say, there's 83 what we call composite restraints, there's 30 shape restraints, there's 2 symmetry restraints, there's an 8-fold and 2-fold symmetry, and there's 30 restraints that are positional here to give you a sense.
So you actually don't require the particle to restraint ratio to be around 1. You can have it much lower.
Actually, we don't worry about that first and foremost, because I hope you can appreciate it from how I described the process. We tried to sample at required precision all solutions that satisfy the restraints. And then what matters the most is how precise is that ensemble of good scoring solutions. That tells you what the precision of your model is, And you also know that that precision reflects whatever the uncertainties in the input are. But the most important precision is the precision of the model or uncertainty of the model. And that is directly estimated from the variation among the independently calculated instances of a model if you also have a test that your sampling was exhaustive or likely to be exhaustive, which one can do in certain ways that I'm not mentioning right now. So that's how we think about it. And then it doesn't matter how exactly you count restraints or even, okay.
Sorry, a quick question. How do you decide what's a reasonable error bar?
Yeah. So that's a very important question, and it depends on the type of information. For example, in the case of localization data, you can see these golden particles that obtained from that are multiple ones for one protein in this image. Each one of them corresponds to an independent electron microscopy, a separate electron microscopy image of a separate nuclear pore complex. The multitude of them is obtained through superposing the individual such images on top of each other. And you can, through a small number of simple steps, argue that a measure of variability of the positions of the gold particles on this image corresponds to the error bar, or a measure of an error bar for a restraint based on these images. So basically the replica idea. You repeat your measurement multiple times, and the variation among the independent experiments is a measure of its uncertainty. It gets a little more difficult if we try to think about what's the uncertainty in the symmetric restraint. But in this particular case, it's not so important because you satisfy all of the restraints better than the scale of the model uncertainty here. And so you say it doesn't matter, right? I'm not claiming from my model more than I'm claiming from my restraints. And so you feel, maybe you could claim more from your model, you get a more precise model, a more accurate model, if you had a better, more accurate idea about the data uncertainty. But you admit you don't, and because you're happy enough with the model uncertainty as it is. So I'm not sure I gave you a very good answer, but it is a big question what the data uncertainties are, or it can be, and depending on exactly the occasion, you are or you are not in trouble in this regard. This is an example, a sample of various other studies, applications of integrative structure modeling that we were involved in, always in collaboration with various experimentalists for generating the data we use to construct models and also to test them. And you can hopefully appreciate the variety of these different systems, different sizes. Some of them are dynamic systems, we model the dynamics of a process. Some of them are multi-state systems, we model the functional cycle. And some of them are just single static snapshots of the system. In fact, most of them are still of that kind. But it's not just one-off. And here's one advantage of integrative structure modeling as opposed to non-integrative structure modeling based on more or less a single method. Because we are using, or we are maximizing the amount of information in our modeling, we are poised to expand the envelope of structural biology in a couple of different directions at least. And some of them are listed here. So we can increase, we hope, the size of the systems that are described. We can, as already alluded, describe systems that exist in a sample that we measure in multiple states, few states, not just in a single state. And in the extreme, we can describe sometimes a dynamic process, not just some snapshots. We have a movie, not snapshots of the movie. And then we can use information from single molecule methods as well as bulk sample methods, which can be very important because there's certain types of information you just cannot get from bulk experiments. And we can use, importantly, information from impure samples. Imagine you can determine structures from samples that are not pure, like crystallography or EM and so on. Chemical cross-linking on a cell lysate is an example of that. And importantly, and now I feel I'm not going to have the time to describe it in the third section of the talk, but still, importantly, we can start adding non-structural degrees of freedom to the structural description. And an example of that would be modeling molecular networks, nodes and the edges between them, perhaps in addition to or even instead of structures of macromolecules. Again, with the integrative mindset that takes information from different methods. So I think these are all kind of justifications for integrative modeling in particular can be a good thing. And in order to facilitate the computational parts of the process, that includes experiment and computation, we are developing in our group, which is computational, an open source software we call Integrative Modeling Platform, or IM in short. We try to document it as well as we can, and it's totally freely available. And we designed it as modularly as we can, so that we can mix and match different representations with different scoring functions, with different sampling schemes, and then analyze the results in different ways as a specific application demands. And I am very keen on advertising this, both to people who just want to use the software for this specific problem, as well as method developers who may be interested in adding a restraint based on a new type of data, for example. And then after that, benefit from everything else that's already incorporated in this framework. So don't be shy in case you are one or the other type of a user, potentially. Let me expand on this idea a little bit. So there's clearly synergies in integrating an effort in this way within a single research group, as we do. But why not take that to the extreme and set up the conditions and the framework such that we can mix and match tools for molecular modeling across the planet, right? If we could do that, that would certainly be very good. And obviously, to some degree, that's already possible. You can, everyone is doing that already, and here's some such tools that people have developed for different aspects of protein structure, determination and analysis, visualization, archival, dissemination, and so on. But to be more programmatic about it, more explicit about it, a group of structural biologists of different varieties have met in 2014 at EBI in the UK, European Bioinformatics Institute in the UK, in order to address, in the context of integrative structure determination, a couple of key questions that are listed here in red. And what we call a white paper was written up with recommendations for each of these issues. And then subsequently, importantly, two so-called working groups were formed, mostly from the attendants, but including others, that addressed in practice the recommendations coming out of the meeting. And without describing too much a lot of work that was done, let me just give you the bottom line. The bottom line is that within a small number of weeks, you will be able to read an announcement by the Worldwide Protein Data Bank in the journal Structure of this archive, a new module of the Protein Data Bank for integrative structures specifically. So it will be the biggest expansion of the scope of the PDB after the EM structures were added, in addition to NMR and X-ray structures. Now you will have integrative structures, and the data on which the structures were based, and the protocols with which the structures were computed from the data, archived for dissemination after validation. And that's really an important step. So, that's the end of introduction, right? But it was a little thicker than most introductions, so I hope it's okay. I would like to illustrate now in slightly more detail the application of the method, of the approach, to a particular difficult structural biology problem, the determination of the NUP82 subcomplex structure. So here's the context, and before I do that, I should now mention, I should have done it already. This is a collaboration that's kind of the core of what we do in terms of inspiration and many other aspects. With Mike Rout and Brian Chait, two professors at Rockefeller University, biologists, mass spectrometrists in the case of Brian, a long-standing collaboration, and there are many students and postdocs that are the most important of which are acknowledged here, and all of them that contributed are acknowledged at the end. Without them, we couldn't do anything, basically, is what I want to say as explicitly as I can. We do the modeling, they do the experiments, many ideas, and all that. The nuclear pore, the NUP82 complex is a part of the nuclear pore complex. You've seen this model already from 2007. The NUP82 complex occurs in multiple copies as part of the NPC. In particular, on this image, one copy is encircled in this red ellipse. It's an essential complex of 4 different types of a protein inside the nuclear pore complex. It's important functionally because it's involved in messenger RNA protein complex remodeling, in particular in pre-ribosome nuclear export. But it's a tough target for structural biologists of traditional kind because it contains disordered, these 4 proteins contain disordered FG repeats, making it exceedingly difficult to crystallize, in fact, so far impossible to crystallize the system, as well as perform electron microscopy with it. And certainly it's too large to do nuclear magnetic resonance spectroscopy. So it's a good candidate to showcase what integrative structure modeling can help with. The schematic of the system composition is shown here. The 4 proteins, in fact, each one occur in 2 copies. So there's in fact in total 8 proteins in the complex. You can see there's some parts of it, some domains for some proteins that have previously determined atomic structures, but much of the proteins are undefined structurally, and most importantly, it's not clear how the components fit together in space. And we applied the 4-stage process. I'll use that's outlined on this slide, and I will go through this slide in some detail, but that will be all I will be saying about the method. It's simply a repetition of the approach that I already introduced to you. Now, just specifically, I'll go through the data sets and representations and maybe a little bit about the validation in the end to give you a better sense of how this works. Gathering the data. What are the data? The first of the 4 stages here. They're listed here. Let's just go through it. First of all, the components. We have some X-ray structures for some of the proteins or the domains in them, 12 domains. We have also comparative protein structure models for some additional domains, as indicated here. And incidentally, obviously we know that 2 proteins cannot be at the same space at the same time, so we have their excluded volume restraints as well. In addition to that, we have, importantly, information about which pairs of lysine residues and which pairs of carboxylic acid side chains, the glutamic and aspartic acid side chains, are close enough in the complex to have resulted in a chemical crosslink of a certain length connecting them under native conditions, as detected by mass spectrometry. So this results into distance restraints, and there's in fact quite a large number of them, in total about 800 or so. And then we also have information that's relatively easy to obtain from electron microscopy, unlike information from single particle electron microscopy reconstruction. It is in fact quite easy to construct so-called 2D class averages, which are these images you see here, showing a projection of the system from averaging multiple particle images. But they are 2D images, they are not 3D densities. It's for that reason that they are much more easily obtained, even when the system is heterogeneous in conformation and or composition. So we had this, and whatever model we constructed better be able to orient such that its projection overlaps with this multiple class averages. We also had information from small angle X-ray scattering experiments that effectively result in a radial distribution function for whatever is in the sample, in this case either individual constructs or, I think in one case there was a binary complex between domains from two proteins that we characterized this way. These are the restraints based on mostly the data and some theoretical, so to speak, sources. Then, with the representation of the components that is implied by these X-ray structures comparative models and lower resolution big representations of parts of the sequences for which we didn't have atomic structures involved, take these components and again apply a version of Monte Carlo, shaking and baking in space, trying to come up with models of complex that satisfy these restraints as well as possible. And in this case, we do it in 2 stages, not worth going into details. It's basically a stochastic Monte Carlo sampling process that will result, in this case, in structures that satisfy enough restraints and turn out to be quite similar to each other, as before for the nuclear pore complex. Now, admittedly, this was only a result after a number of years of effort and after a lot of expansion of this input information was done through a collaboration with our experimentalist friends based on initial failures of the modeling that themselves were based on smaller and perhaps noisier input data sets. So there was very much an iteration through the scheme. Nevertheless, at some point, we were able to show that there's only one major solution that satisfies most of the restraints within the error bars. And we were moreover able to to show that this solution satisfies both the restraints used for modeling as well as restraints not used for modeling or data not used for modeling, such as positions of green fluorescent protein in the GFP tagged constructs of the complex. And as I mentioned, the sex profiles also, you can see that were not used in modeling, describe the arrangements of parts of the system quite nicely, as illustrated on this plot here. So just to give you a sense, so we again, in terms of validation here, and it's a topic on its own, an important one, we work on this hard enough until we get as often as possible a single cluster of structural solutions, which we then validate in various ways, including based on how well they satisfy the data used to compute the models, as well as data not used to compute the models.
I'm sorry, a quick question.
Yes.
I wasn't clear in the initial random configurations, the domains which do not have a predetermined structure, you represented them by beads?
Beads, yeah, these beads. So this would be maybe 20 residues in this bead. We just don't know. What can we do?
Why wouldn't we, I'm not a structural biologist, but why wouldn't you use strings for coiled?
We want to be as conservative as we can. So again, it's a big topic, and there is work to be done still on optimizing the representation based on what we know about that particular part of the system. Admittedly, and we are engaged on that. But at this point, what we do is just use a very low resolution representation, such as a sphere of, say, 20 residues. Consecutive such segments will be represented by consecutive beats that are required to be close in space if they're part of the same polypeptide chain. And then excluded volume is applied to these beats relative to all the other components in the system. And sometimes you may know, you also do consider them when you're fitting, I guess, these models against projections from electron microscopy. And you just have to be appropriately soft when you are required in terms of quantifying a match between a model and a restraint, considering the resolution of the representation. It's a big topic, and I could probably have multiple lectures on that. What's an optimal representation? How is that related to the information available? How is that coupled with the precision of sampling, as well as the precision of the final model that you can claim through some analysis of the final model ensemble. But maybe for today, this will be it.
So the data for the validation, that information was not available earlier, or?
We proactively decided that we will leave some information aside,
So is it equivalent to an
only
R
for
free set?
validation. I'm sorry.
Is it like an R free set that you use for?
You could formulate this process a little more systematically along the lines of the R3 criterion in crystallography. It is not quite as straightforward because it's a little more difficult to mix and match different types of information in this way, but you can think about it in exactly, it's a special case of cross-validation, if you will. And there's no particular reason why we pick this or that to leave out. We could also do a computation with all of the data in the input and estimate the precision through some kind of jackknifing after the fact. There's a little still haphazard way, experience-based way. When exactly do you get the data? What will take less CPU time to include in the sampling a million times as opposed to just once at the end, and so on. Yes.
So you can presumably go back and see which source contributed how much to the final source of introduction of information
Yes.
contributed how much to the final structure, right?
Yeah, you can, more specifically probably what you mean is you can compute how much the final structure changes or not if you omit a certain data point from your input. Is that what you meant?
No, I actually meant whether certain, so X-ray, the crystallography should contribute more probably to getting the final structure. Some are less important.
Well, but you have to define what you mean by contribute more or less.
Oh,
I
no,
think the only thing you can say is how much the structure changes
Right,
or
that's
not
true.
if
Correct.
you omit
Right, that is right.
information
Yes.
you want to
You
examine.
can, right? So, and then can you then go back and say that maybe this instrument was wrong by so much?
Yes. Yeah. Yes.
That
Absolutely.
maybe the EM machine is wrong.
So
by-
now you're raising the point. So this was formulated. This particular modeling problem relied on almost fully Bayesian scoring function, and therefore, what you can say is that this final step doesn't validate only the model, but it actually validates also the data. In fact, it validates the data through the model by quantifying how consistent or not is any single data point, any subset of data points with the remainder of the data points. That's really all you can do in terms of validating the data. But in the Bayesian approach, that's very much a part of it if you wish. And yes, the fact that we were able to compute a three-dimensional model, a single three-dimensional model at some precision, that satisfies all the data, validates not just that model, increases your confidence not only in the model, but also in the data themselves. There's no guarantee that there is a single three-dimensional structure satisfying data from totally different experiments with physically different samples. And when you do find that's the case, you have an increased confidence in everything, the data, the model, and your conversion of the data in the model.
So can I just,
Yes?
I think this may be related to what you just said, but what do you do with conflicting pieces of information? Do you choose one over the other? Do you discard the
Yes.
both? Or?
So first of all, you hope that there are none, or that there are a very small number of them. For example, for chemical crosslinks, there's been prior studies that show that about 3–4% of chemical crosslinks are what can be called false positives. They are inconsistent with X-ray structures in controlled cases. You say they are mistakes in the measurement, they may be a consequence of temporary fluctuation of the protein structure, they're just not consistent with a single structure, 3–4%. Controlled cases, a number of times, different authors agree on that. So, if we now find that our models at the end violate 3 or 4% of chemical crosslinks, we're not going to sweat it. Right? We're going to say, we did as much as we could. Our model satisfies as much of the chemical crosslinks as we can reasonably expect, given the uncertainty in the chemical crosslinking experiment. So you want to do the same sort of thing for each type of information independently. When that's the case, you're done. Your problem is a nightmare. You know? What happens when that's not the case? When, for example, the best you can do is satisfy only 90% or 80% of the chemical crosslinks. What do you do then? What are the possibilities? The first one is, well, the chemical cross-linking experiments in this particular case were particularly bad. That's not very popular usually as an option, but mathematically you do list it as a possibility. The second option is a class of them that have to do with modeling. And remember, there's three kinds of modeling, three aspects in modeling: representation, scoring, and sampling. Any one of them could be wrong, absolutely. It often is. A mistake in representation that would result in 10% violated crosslink could be perhaps using too many residues in a bead, using beads that are too big. So you freeze the degrees of freedom too much, you don't let the model relax enough to satisfy the crosslink. As an example. Another big mistake that can be made is who says that the system exists in a single state. Either in one experiment or especially in different physical states that are usually required for different experiments, all of which you originally wish to satisfy with a single state in your model. So what you can then model a combination of two states, or 3 or 4 and so on, to satisfy the data and do so-called multi-state modeling, which we can do in some situations, but obviously it's more difficult in a sense that you have more degrees of freedom that need to be informed by the data, so you need more data. But at least in principle, it's the more accurate way of representing what the problem actually is. You can have your sampling that fails, not just your representation. If you don't do enough sampling, well, maybe there are solutions, single state, that satisfy 96% of the crosslinks. You just didn't generate them, and your computer is too small or whatever. And finally, scoring. Yes, maybe your weights or functional form of how you express information, not just about the chemical crosslinks, but everything else that's in the scoring function, is not accurate, and there might be some mistake there. So, other than the representation, right, even if representation is correct, you could still make a mistake with scoring. So these are options, and now at least you know that you have a problem of one of these kinds, and you can go, or one or more of these kinds, and you can go systematically through it, perhaps acquire more information, perhaps do some control calculations and so on, double checks and so on. And you are you are better off, even though you still haven't finished it. Okay, so I think I'd better move on. But I love this interaction, much appreciated. Thank you. So that was the process in this case. And this is a result. The first result on the left, you see how the 4 proteins, 4 protein types, 2 copies each, 8 proteins in this are arranged for the system. On the right, it's just a movie of the same thing. You can see there's a symmetry somewhat, but not entirely. And one can analyze this, but we won't right now. I'm just showing this result. That's how it looks. What I'd like to show you is the sort of things that you can do once you get a structure with an integrative approach. And the very first step that we did in this case was to expand the structure, add other components to it. Because we know we have a much bigger system at hand. This is a subcomplex. The NAP82 complex is a subcomplex of a bigger nuclear pore complex. And in fact, it's possible to do chemical cross-linking experiments, not just with the natively isolated NAP82 complex, but with some components of the NPC complex present in solution as well. And it turns out that one can observe chemical crosslinks between the NAP82 subcomplex we just characterized structurally and proteins in yet another subcomplex of the NPC called the NAP82 complex. In fact, there's quite a number of such crosslinks as you can see here. And therefore, we can apply another round of integrative structure modeling to position these 2 subcomplexes with respect to each other. And that's the NAP84, that's the NAP82 for now 7 plus 8, 15 protein complex that you see a movie of on the right. So we're bootstrapping up. And then we can go rapidly through this. Think about how you might validate this with functional experiments to do with mRNA export out of the nucleus, since we know that the NAP82 complex is involved in this export from the nucleus. We can speculate, for example, that the proteins involved or the domains involved in the interaction between the NAP82 core complex and the NAP84 complex, which is the scaffold of the nuclear pore complex. These interactions here will be essential for the mRNA export function of the nuclear pore complex, and therefore if we mess with these interactions through domain deletion experiments in certain ways, through positive control experiments, negative control experiments, we can gain additional confidence in this structural model, and that's what this shows. And I'm sorry, I'm not going through the detail because I'd like to get to the end without going beyond the time allocated too much. Moreover, we can also fit a model of the NAP82, NAP84 system, all 16 copies of it, into the low resolution model of the nuclear pore complex, a model that defines only approximate positions of these subcomplexes, but no atomic structures, no orientations. And that's what's done on this slide for both the yeast nuclear pore complex and also the human nuclear pore complex on the right. And in both cases, it's possible to obtain a fit consistent with this electron microscopy-based density map for the human and the what we call localization probability density map from integrated structure modeling of the yeast and NPC on the left. And then finally, we can, again without belaboring the details, we can use this pseudo-molecular model of the NAP82 complex in the context of the NPC to, in much finer detail, rationalize how the export of mRNA protein particles occurs, and we can rationalize quite a number of mutations and their impact on the function that are known for yeast and human. Many of these are disease associated. We can suggest specific further experiments that hopefully would increase our confidence in this model further, having a structure in hand. That's what I wanted to say is it's gratifying when you don't stop just with a structural model after modeling, but you keep going towards perhaps increasing the scope of a model or adding something to our functional understanding of the system. Not mentioned are certain evolutionary insights as well that we described in this paper, but I'm not showing slides of. And I will have to, by necessity, skip the third part of the talk because there's no time for it. It was about formulating modeling of molecular networks with the integrative mindset, benchmarking it, and showing its application to a discovery of a new metabolic pathway in a certain organism. Here, Haemophilus influenza, and validating it experimentally. The hope was that this would further demonstrate the advantages of thinking about modeling, especially integrative modeling, in a very broad sense. But you have to take my word for it. This is a summary of a couple of points I'd like to underline again, and that is that determination of assembly structures that I did discuss, as well as mapping of networks that I haven't really described, benefits greatly from including all available information, which you can only do through modeling. I was advertising our particular modeling program to support the computational aspects of this process, and I illustrated it on determining the structure of the NAP82 subcomplex. And then I would like to thank quite a lot of people. On the left, you have our group members who are currently contributing to integrative modeling specifically, and you have a list of former members who contributed to integrative modeling specifically. There were other members like me who contributed to other aspects and not listed here, but we really benefited from quite a large number of people over the years. I'm telling you about 10-year plus 15-year effort. We also, as I already indicated, are constantly benefiting in so many ways from various collaborators on the experimental side, many of which are listed here. And then in particular, we are involved in the protein data bank effort on bringing the integrative structures to the community with everything that that entails. And I'd like to highlight in particular Helen Berman, who's now totally dedicated to this aspect of the protein data. She previously led the American arm of it, but now she's dedicated to this aspect. And also a number of other scientists who are also helping us here. And specifically for the nuclear core complex work, the NAP82 complex project in particular, S.J. St. John Kim was the key modeler on our side. He benefited from help from other members of the group, but as I keep saying, it would be totally impossible without the data and ideas and so on from our experimentalist collaborators. Here's molecular biologist, mass spectrometrist, electron microscopist, X-ray crystallographer, and the small angle scattering groups that were essential for what we did together. And this was an equivalent acknowledgement for the molecular network mapping project, but I guess I didn't get to talk about it because your questions were so numerous and so good. Not my fault, right? Thank you.